people are the most beneficent and merciful. Here I am, Muhammad Jamal Khan, lecturer in botany, University of Education, Lahore. Dear students, Assalamu alaikum. Today we will discuss a topic that related to our elective subject, that is microbiology. So this is our lecture number 9. In this we discuss about the scanning probe microscopy and confocal microscopy. So actually, scanning probe microscopy is the advanced form of electron microscopy that is further classified into two forms that we discuss later in this lecture. So here we have scanning probe microscopy, advanced form of electron microscopy. The other one is the confocal microscope, that is also the form of light microscope. So in scanning probe microscopy we have further classified into two forms. One is scanning tunneling microscope and the other one is atomic transmission microscope. So we discussed this earlier in this lecture. And in light microscope, in which we discuss its type, that is confocal microscope. So now we start from scanning probe microscopy. So simply, as you know, the light and electron microscopy have become quite sophisticated and a very advanced, you can say, form of microscope or new microscope. Although the electron microscope is the most powerful microscope in this world, but in addition to this, a new class of microscope called scanning probe microscope that measures surface features by moving a sharp probe over the object surface. So this scanning probe microscope basically invented in 1980 in the form of scanning tunneling microscope. That is the example of scanning probe microscope. So the power of scanning probe microscope is so much greater than electron microscope, and only a simple sample is being used in this microscope as compared to the sample that is prepared for electron microscope. So due to its high resolving power, this microscope maps the bumps and the valleys of surface on an atomic scale. So actually in this modern era we have two types of scanned probe microscope. One is scanning tunneling microscopy and the other one is atomic force microscopy. So first of all, I have a tutorial for you for scanning tunneling microscopy. In this tutorial one explain a very descriptive manner regarding the working principle and the importance of scanning tunneling microscopy. So here we have the tutorial of this.
Now, scanning tunneling microscopy is one of the major advances of the change in electron microscopy, and it's a kind of different feature of microscopy that we use. Because up till now we have talked about the microscopy with the light source, right? They are called the light microscopy or bright field microscopy, I can say. Then we have also seen using electron, we can actually look at very high resolution images using the scanning electron microscopy as well as the transmission electron microscopy. So it's electron as a source. Now in this case of scanning tunneling microscopy, we are also using electron as a source, but remember here the electron that we use in electron microscopy, that electron behaves like a particle, just like the classical phenomena of physics. But the electron that we use in scanning tunneling microscopy behaves like a wave. So this is called the quantum mechanics. So the way that we use, the idea, the principle that we use in scanning tunneling microscopy is about with the quantum tunneling effect, or with the quantum physics, the law of quantum physics, right? Because the electron can behave like a particle. When an electron can behave like a particle, let's say we have a barrier in both the cases. But when the electron behaves like a particle, just like the classical physics suggests us, in that case that electron, let's say, that electron will go and hit to that wall and bounce off and release. So this is a reflection if the electron behaves like a particle. But this electron, if it behaves like a wave, this electron has the capability to pass through that wall. That is the idea. So that is called as the quantum tunneling. So that's what we talk about in this video. So this is the principle that is the core foundation of quantum tunneling microscopy or scanning tunneling microscopy. Remember, STM, scanning tunneling microscopy. Why the term tunneling here? Because of the use of quantum tunneling effect. Now the quantum tunneling effect is telling us the classical picture is electron can go and attach to the energy barrier and hit the energy barrier and can return. But in case of if they behave like a wave, that wave can pass through that energy barrier from one side to another side. That is possible. That is the quantum tunneling effect. We use this quantum tunneling effect here. So the idea here, what we do in the scanning tunneling microscope is that obviously we have a sample, right? We have a sample, whatever sample let's say we have. That sample majorly we use in this case is not always living sample or not even living samples. We use this quantum tunneling effect or the scanning tunneling microscopy to understand the surface of different atoms, the surface of different, let's say, different molecules that we see, right? Different metals. We can see the surface of graphite, we can see the surface of gold, surface of silver, and all this, the surface of them. So how they are arranged, how they are organized. So it's not only important from the perspective of biology, but it is more important in the perspective of chemistry and physics. Okay, and many more, geography also, because geology we often use scanning tunneling microscopy. Now the idea here, we have the specimen. This is the specimen, and what we also have here is the structure called the tip. The tip is nothing but it's made up with metal with high conductance, and this tip is responsible for releasing electrons from there, right? So what we actually do, we connect these two things. Both of them are electron carriers. So we actually connect the tip with the sample in some way, right? So if we connect it with a particular voltage, remember, if we add a particular voltage between this tip and the specimen, electric current start to flow between them. That is the idea. Due to this voltage between the tip and sample, current waves start to flow from the tip to the sample, or from the tip to the specimen, right? So this is the idea. Now the idea here is the amount of electricity that is generated and that is formed between the tip and the sample, it is indirectly proportional to the voltage. So if you apply higher voltage, the electrons that will be released will be low. If we use lower voltage, the current flow will be high, right? So they have indirect proportionality between this voltage and the current flow, right? So if you look at in this image, we can see it pretty clearly that what we use here, the tungsten tip here, and in this case you can see this is the tip of the atom with a positive charge, and here is the sample atoms, negative charge, and we create this voltage there, attach these two things to pass some light, pass some current between them. Here you can see the voltage arranged there, right? So once you do that, the idea now is to detect the surface structure of the specimen, which is this one, right? So how could you do that? Now here is the idea. Now let's say this is it, and the sample, let's say this is how the sample looks like. For example, the surface of the sample looks like ups and downs and all the way like that. Now here's the idea. We have this tip. Now the tip is attached with a rotator which can move from one place of the sample to other place of the sample, right? So during that process, we can use two important techniques to find out the surface structure of the sample. One of the technique is termed as the constant current. Another one is termed as a constant height. So constant current and constant height. The idea in case of constant current is, let's say we bring this tip here. Say this is the tip, for example, say tungsten tip. Okay, now the idea in this case is, so the idea here is that this tip is providing that current because of this voltage gradient, because of this whatever voltage gradient is present, it is transferring some of the electrons there. So as it is transferring some of the electrons during this process, so there will be a particular current that will be generated between the tip and the sample's surface, right? Now as you can see, there are two factors. One is the amount of current, another one is the distance between the tip and the surface. The idea here, if the distance between the tip and the sample surface is close, I mean they are very close to each other, in that case the current flow will be more because the distance is less. So if the distance is less, the current flow will be more, right? And the distance is high, the current flow will be less. Again, indirectly proportional in this case. So indirect proportionality again. So the idea again in this case, what we see here is that the tip, what we want to maintain is a constant current, right? So if we want to maintain a constant current, in that case we should have a constant flow of current irrespective of the distance that we see. So we need to bring this, we need to position this tip in such a way so that current remains constant throughout the scanning period, right? But the surface area is not even, it's uneven. So the idea here, the distance between the tip and the surface area point here in this case, let's say this is point A, the distance between point A and tip here is small and there is a particular current. Now once this tip moves a little bit and the tip is now in position somewhere here, and as you can see the distance between this tip and the position B of the surface is more. So accordingly the current flow will decrease. But here we want to maintain a constant current. So to maintain the constant current, this tip will come down a little bit to compensate the distance between the tip and the point of the surface to maintain that constant current. Similarly again, when it comes down to this point of C, the distance even becomes more. So the tip should come much closer to again maintain that constant current. So to maintain the constant current throughout the surface, like uneven surface like this sample, this tip needs to come down sometimes and go up sometimes. So in this way, whenever it is coming down, whenever it is in close proximity with the sample specimen, in that case that data is fed to the detector. Detector record that the specimen is in depth there, and wherever there is a height up there, so the tip will go up. So again in that case it will record the data that yes, definitely the surface is higher, that's why the tip goes up. So whenever tip goes up in the detector, it detects the higher surface. Whenever tip goes down, it detects the lower surface. In this way we can actually construct this image by looking at the detector, kind of the similar image that we see in case of the sample. That's how we can detect using the constant current. And we can also use the constant height method. In that case, what we do, in that case we maintain the height constantly. The same thing is going to happen. If we maintain the height constantly, this tip is going to be present as it is and throughout the scanning process. But let's say here, once this tip is present to the point A, which is pretty closer, so the current flow is high in that case, very high current flow. Now just after that, when the tip is now becoming to the point B, the current flow will be less. So what we observe in detector is a change in the current flow, because we maintain the height constant, so there won't be any change in the height. The only thing will change in this case is the current flow. So sometimes we see high current flow, sometimes we see low current flow. So if we see, now let's say if we see the high current flow in this case, let's say in point A current flow is very high, then again it's going down and low, then again slightly high, then low, then slightly high. So again we can construct the surface structure pretty easily. So these are the two methods of calculating. So this is the principle. As you can see here, this is the STM tip, and again these are the electrons that are coming out from the STM. This is the voltage gradient, plus here, minus here, and as it is moving in different surface, let's say the surface area is changing like this, you can see, as you can see here, difference in the graph, as you can see in detector. So detector can detect this change in distance or change in the voltage, change in the current, right? So by looking at this, we can tell what's going on. And the idea here is this tip will migrate from one place of the sample to another place of the sample, from scanning that sample from one to another place, right? So what it uses here, it uses the property of electron as a wave for this electron to be inserted to the surface of the sample so that the current flow is possible, right? So that's how the whole process of scanning tunneling microscopy actually work. And this is the instrumentation of scanning tunneling microscopy. As you can see here, let me take another, yeah, as you can see here, this is the probe, this is the tip, tungsten tip usually, this is the sample, the voltage is applied between this tip and the sample, and it is being tunneled with the amplifier, because the thing is the energy, I mean whatever surface current it generates, that current is pretty low for the detector to detect. So what we need to do, we need to amplify that signal. So for amplifying that signal coming out from the tip, we require this tunneling current amplifier, because this tunneling current amplifier will amplify the current that is coming from this tunneling effect between the tip and the sample, and we get that, and we have a control scanning unit which controls the scanning property, how this tip will move from one place of the sample to another, and then finally we can record this data using a CPU or processing unit. Okay, and this is in action how the process works, as you can see from different angles actually, but the idea here, you can actually construct this kind of beautiful images of the surface by looking. And this is the actual scanning tunneling microscope. That's how it looks like. It not at all looks like any kind of microscope that we've seen previously. So it's a microscope because it helps us to understand the structure in very fine level, because the distance between the tip and the sample is kind of in angstrom level. So it's very good actually in resolution and all these things. As you can see here, it's even more like 50 nanometer range. It's giving us that much of the detail of the structure in different layers. It's very good, but we can use it for live imaging also. We can use it for, let's say here you can see the cells, and we can also see some cilia coming out from cell surface. So we can actually use it in different purposes. But the use of scanning tunneling microscopy is not limited to biological applications. It is vastly used in many different fields of science.
So here I have the image of DNA double helix structure that we obtained from scanning tunneling microscope with approximately 3 tons shown. So it's a high magnification power image that we get from scanning tunneling microscope. So here we have the second type of scanning probe microscopy, that is atomic force microscopy, or AFM. So the AFM is the more advanced form of scan probing microscope, and it moves a sharp probe over the specimen surface, and while keeping the distance between the probe tip and the surface constant. So it does this by exerting a very small amount of force on the tip, just enough to maintain a constant distance, but not enough force to damage the surface. So basically the vertical motion of the tip usually is followed by the measuring the deflection of a laser beam uh, that strikes the lever holding the probe. So basically the tip that is used, this tip that is used to probe the specimen is attached to the cantilever. This one is a cantilever. So as the probe passes over the hills and valleys of the specimen, if this specimen possessing some sort of hills and valleys, the cantilever is deflected vertically, so it's moved vertical up and down movement, shows up and down movement. So a laser beam directed at the cantilever is used to monitor these vertical movements, and light reflected from the cantilever is detected by the photodiode and used to generate an image of the specimen. For further illustration, I've attached animation of atomic force microscope here.
Atomic force microscope. It lets you see things that are very, very small, all the way down to the nanoscale. How does an AFM work? An AFM uses a cantilever with a very sharp tip to scan over a sample surface. As the tip approaches the surface, the close-range attractive forces between the surface and the tip cause the cantilever to bend towards the surface. However, as the cantilever is brought even closer to the surface, increasingly repulsive forces cause the cantilever to bend away from the surface. An AFM has a Z scanner that moves the cantilever up and down, an XY scanner that moves the sample back and forth underneath the cantilever, and a position detector that records the bending of the cantilever. The position sensor works by tracking a laser beam that is reflected off the flat top of the cantilever. Any bending in the cantilever will cause changes in the direction of the reflected beam. The position detector then tracks and records those beam changes. Using the detection method explained earlier, an AFM images the topography of a sample surface by scanning the cantilever over a region of interest. The raised and lowered features on the sample surface influence the bending of the cantilever measured by the position detector. AFM generates an accurate topographic map of the surface by using a feedback loop to control the height of the tip above the surface. There are two major methods of AFM imaging: contact AFM and non-contact AFM. In contact AFM, the cantilever scans across a sample with its tip touching the sample surface. Because the tip is in contact with the surface, strong repulsive forces cause the cantilever to bend as it passes over the surface features. The advantage of this method is the relative simplicity of its technology. However, there are disadvantages. One disadvantage is that the sample can be damaged by the gouging action of the sharp tip. Another disadvantage is that while the tip is gouging away at the surface, it also grinds down the tip, which reduces the quality of your image. In non-contact AFM, the cantilever oscillates just above the surface while it scans, but the tip does not touch the surface. A precise, high-speed feedback loop is used to keep the cantilever tip from crashing into the surface, thus retaining tip sharpness and leaving the surface untouched. As the tip approaches the surface, interactions between the tip and the surface cause the oscillation amplitude of the cantilever to decrease. The feedback loop corrects for these amplitude deviations and constructs an image of the surface topography. Non-contact AFM has many advantages over contact AFM. It preserves your sample surface because the tip and surface are not touching. By prolonging the sharpness and lifetime of the tip, it allows for high-quality imaging over many scans. This also brings down the total operating cost of the instrument, as tips are expensive to replace. Atomic force microscope, or AFM, is a powerful tool that enables you to see, explore, and obtain measurement data from your samples at the nanoscale. For more information, please visit parkafm.com.
So here we have some of the application of atomic force microscope. First of all, the study surfaces. So the scanning microscope, the atomic force microscope, can be used to study surfaces that do not conduct electricity well. The next one is the visualize proteins of bacteria. So the atomic force microscope has been used to study the interaction between the E. coli and GROL chaperone proteins. So to map plasmid by getting restriction enzymes bound to specific sites. So the next one we have studying the DNA bases. The atomic force microscope has been very useful in studying the DNA bases because it enables investigators to distinguish between bases such as adenine, guanine, from differences in their electron density rates. Uh, the other application of electron microscopy is used underwater. So it has also been used underwater to study chemical reaction at living cell surfaces, which help to confirm chemical analysis of the cell wall material. It also enables us to produce images that can measure forces. For example, the force needed to unfold a protein located in the membrane. And the last one, it determine the flexibility of polysaccharide molecules. So that is to know how far it can elongate before it ruptures, and the information is important in studying attachment of adjacent cells to form aggregations such as colonies and films, or for the ability to attach to the host. So the next form of light microscopy is the confocal microscopy, or confocal scanning microscopy. So here we have the simple introduction of this microscopy. First of all, as you know, most of the biological specimens are three-dimensional. So when we observe these three-dimensional objects through a traditional light microscope, light from all areas of the object, not just a plane of focus, into the microscope, and are used to create an image. And such image that is produced by this is mostly very fuzzy. So this problem has been solved by the development of confocal scanning laser microscope, or simply we call it as confocal microscope. So here we have the simple working principle of confocal microscope. First of all, the confocal microscope basically uses a laser beam to illuminate a specimen, and usually one that has been fluorescently stained. Uh, it means the specimen that we used, we stain it with any fluorescent compound. So the major component of the confocal microscope is an aperture placed above the objective lens, so which eliminates stray light from parts of the specimen that lie above and below the plane of the focus. This statement is further elaborated and illustrated by the tutorial that is attached below in this lecture. So the only light used to create the image is from the plane of focus, and a much clearer and sharp image is formed. So here we have the tutorial.
Microscopes. This is a Zeiss confocal microscope. Here you can see the main part, the main confocality lies in this scan head. So the scan head holds a pinhole and the detector. So this is the important part. If you get rid of that, the rest of the microscope is nothing just like a epifluorescent microscope. And one of the reasons to do confocal microscopy is to get these beautiful images. But that's not actually true. Here are two same images, which are taken in an epifluorescence microscope and a confocal microscope. Clearly, you can tell in epifluorescent microscope, there are a lot of backgrounds, as if the image is like fuzzy and blurry image. Also, another image, you can see the same. The confocal image is way more sharp and crisp, and the resolution is better in case of the confocal microscope image. And the reason being, confocal uses laser, whereas epifluorescence uses normal light of single wavelength. So since the laser has a property of coherence, the laser can be focused into a very narrow point known as laser waist, whereas the waist is way bigger in case of epifluorescence. So the chance that less amount of fluorophore would be activated at a time in confocal is high. But in case of epifluorescence, quite a lot of fluorophores in the surrounding regions would be also activated, and that give rise to that blur. Now, here is a fluorescence microscopy diagram. So we give a excitation light, and let's say our sample has FITC or GFP, anything. So we are sort of exciting it with a 488 wavelength light, and we are collecting the emission in a camera. So the fluorophores that we are targeting would be kind of excited, and we would collect the emission light. But the fluorophores of upper and lower plane of that image would be also excited, and also it would enter the camera. That would result in a blurry image. Whereas in a confocal, in the same excitation paradigm with a laser, we are collecting the emission light. And since there is a pinhole, the pinhole is confocal with the specimen plane. That means the specimen plane and the pinhole are in same focus. So only focused light can pass. So here is another light ray which is coming from above or below plane of that imaging plane. So it cannot pass through the pinhole and reach the detector. Thereby we are not getting the stray lights, and that's why the quality of the image is actually increasing, also the resolution. Now we would talk about the optical sectioning and Z-stacking property of the confocal system. So confocal system comes with a motorized stage. So the distance between the objective lens and the stage could be altered in real time, and thereby it can at a time image several focal planes, and after that all the focal planes could be merged to reconstruct a 3D image of the object. Now if I give you an example, it would be way clearer. So you can see here the objective is moving up and down, or the stage is moving up and down. Main point is the distance between the objective and the stage is changing, and thereby you are going through the image plane. You're going all around the image plane and imaging all the planes. Later a 3D reconstruction of the image could be possible like this. So we are actually not physically sectioning, whereas we are at a time imaging from a particular focal plane, and we are collecting light from a particular focal plane. So that gives rise to these beautiful images. Another important point, and most important point to get good confocal images, is to how to optimally adjust gain and offset. First of all, what is gain and what is offset? So gain means, gain is simplistically, gain is a parameter changing which we can increase or decrease the signal. If we increase the gain, simply the signal will increase. Later while we would discuss the detectors, we would understand the basis of increasing gain. But increasing gain means increasing voltage across the PMT electrodes. Offset means addition of negative or positive voltage. Now if I give you an example, these things would be more clearer. Now here you can see this is the baseline photomultiplier signal. So even if there is no specimen under a microscope, there is some amount of signal coming to the detector. Due to some thermal emission of electrons, due to other reason, there are some kind of signal. Now by giving a negative voltage, you can make that residual signal very close to zero, and then you can increase the gain or increase the voltage across the PMTs, across the electrodes of the PMT. You can increase and boost the signal by changing gain. Now here is a image where you can see the gain here is increased too much. That means way too much voltage is injected. That's why you are seeing everything is saturated. So this kind of paradigm is not proper. Another image you can see here, it is pretty much offset, but the gain is not increased properly. That's why still the features here that we wanted to image are still dim. So gain and offset need to be properly adjusted such that we get a proper dynamic range of detection. And in a moment I would discuss what is dynamic range. Simply, in an image, if we increase the gain, it would increase the signal, but at the same time you see the image looks more grainy because the background signals also increase while we increase the gain. Now, why does anyone need to adjust gain or offset? So in order to utilize the full dynamic range of imaging, a person need to adjust the gain and offset optimally. Here you can see there is an image, and this image is quite proper. An image that has all sort of gray values would be called a good image. So if you don't understand this image, I have another image for you. In this image, the black things or black or bluish kind of pixels are actually low valued pixels, whereas a yellowish pixel is very high value pixel. Now if you look at the image, you would see all sort of colors are present. It's not that one color is present more than other colors. This kind of all colors are blended properly. So all sort of gray levels are present. That means we have utilized the whole dynamic range, and we have kind of imaged for all the values and captured all the features of it. Now I would show you three comparison. Here the image looks too saturated, because if you look at these lookup table, you can see there are too much white into it. That means the image is too saturated. In order to improve the image quality in this case, you need to reduce gain or laser power. Here the dynamic range is kind of proper because all sort of gray values, all sort of range of pixel intensities are captured in this image. Whereas this image has way too low signal because you can barely see all the features inside this image. So we are missing a lot of information from this image. Now I would talk about another important feature which also is important for resolution of confocal image is the digital zoom. Apart from the normal zoom, apart from the normal magnification, that means for example we are increasing the magnification from 40x to 60x. Apart from that, there is something called digital magnification or digital zoom. So what happens in digital zoom? Let's say the laser is scanning, we are at 40x and the laser scanned the whole field, whole cell depicted here. When we increase the zoom, then the laser would scan for a small feature in the cell. So it's simply scanning the less area. But the image that would be produced in both the cases have the same size. So more feature would be kind of displayed in case of when we increase the zoom. For example, let's say there are 3 pixels very close enough. Now if we increase the number of pixels by projecting the image into a bigger frame, then there are more gaps between it. That increases the resolution. In other words, in case of increasing zoom, we are not changing the imaging size, but a small subset of feature is projected onto the same number of pixels. So pixels per feature is more, such that the image quality gets better and also the resolution gets better. Now for example, here is the image in 40x. Now once we add a 3x digital zoom, you can clearly see the image quality has improved and the resolution also looks way better. Now we should talk about the scanning modes and the type of scanner used in confocal microscope. So definitely we use laser light sources for confocal microscopy. So laser kind of scans in a small portion. Now question is how the laser rasters in a sample in a small spot. So the answer is there are several kind of scanners that are used in the confocal microscope, which are galvanometer based scanner and resonance scanner. Why different type of scanners are used? Galvanometer based scanners use 2 mirrors which are perpendicular to each other and they are vibrating while there is a change in electrical field. So galvanometer scanners are slow scanners. They can scan at a rate of 1 to 5 frames per second. Whereas the resonance scanner are pretty fast scanner and they can scan 30 frames per second. So in order to catch fast dynamic live processes, a resonance scanner would be a better option. These days there is hybrid scanner which employs both galvanometer scanner and resonance scanner. So those are known as also hyper scanners or hybrid scanners. Next we would talk about the detectors used in confocal system. Confocals mainly used photomultiplier tubes or PMTs. Now here is the basic outline of a PMT. So there is the incident light. Incident light is the emission light from the fluorophore and it would hit a photocathode. Now this photocathode is generally made by multi-alkali or sometimes alloy like gallium arsenide phosphide. Now different material can be used to build this photocathode. Depending upon the difference in the material used, the photocathode sensitivity could be also changed. However, when the light hit the photocathode, some electrons are ejected. That electrons are actually going throughout successive anodes and ultimately reaches the end anode. And there is another digital to analog to digital converter which is converting these signals and digitalizing this signal, and thereby the image is reconstructed point by point. Now here is an example of two different kind of detector that could be used. In case of multi-alkali detector, the problem is when the electrons are bouncing back and forth in the dynode arrangement cathode, the problem is there is a loss of electron, and that means some amount of signal is also lost. But in case of a hybrid detector, special arrangement of the detector ensures that the loss of the electrons are minimized. So that's why hybrid detectors are better. Now here I would talk about what determines or what factors determine the image quality of a confocal microscope. Simply the pixel dwelling time, laser intensity, pinhole diameter, gain and offset, fluorophore quality and detector quality, all these factors are important in determining the confocal image quality. So I would briefly go over through these points because in the next video I would discuss these points in details. So pixel dwelling time means how much the laser is dwelling on a particular point. So if the laser dwells too much time, there is a possibility of photo bleaching. So you have to optimize the dwelling time. Laser intensity is another important factor. If the laser intensity is too low, the features won't be illuminated. And if the laser intensity is too high, then the features would be saturated and the dynamic range won't be fulfilled in the image. The pinhole diameter is another important thing. If the pinhole diameter is too narrow or too less, then what happened? A lot of signal would be cut off and the image would be dim. But if the pinhole diameter is more, then what would happen? The out of focus light can get through this pinhole. Now also gain and offset are other parameters that are important determinant of image quality, because I have already shown you with examples that if you don't adjust gain and offset properly, the image looks horrible. And other than that, fluorophore is the most important thing. If the staining or the fluorophore quality is not good, or say the fluorophore is bleached, then you are never going to get a good image in confocal microscopy. After that, detector quality is important. Now we have to ensure that we are always imaging in a linear range of the detector in order to get good images. Now definitely the hybrid detectors or gallium arsenide phosphide detectors are way better than the multi-alkali detectors. That's why choosing the proper detector for your system is also very important. Now if you like my video, give it a quick thumbs up. Don't forget to like, share, and subscribe. Thank you.
So here we have the applications of confocal microscope. First of all, with the help of confocal microscope, we are able to study the biofilms without disturbing them, which can form on many different types of surfaces, including in developing medical devices such as hip joint replacement. So it is difficult to kill all cells in a biofilm. So this makes them a particular concern to the medical field because formation of biofilm on medical devices has result in infection are difficult to treat. So second one, confocal microscope help to obtain the three-dimensional images of entire cells and cellular components. In addition to this, it also helped to evaluate cellular physiology by noting the distribution and the concentration of substances such as ATP and calcium ions. In the end, it also enables us frequently the specimens that are first stained or tagged with a fluorescent dye by using certain fluorescent tags that actually bind specifically to given protein or other compounds. So the precise cellular location of that compound can be determined. So in some cases, multiple different tags that bind to specific molecules are used, and each having a distinct color. So this is all about for this lecture. I hope you get well understanding regarding confocal and scanning for microscopy. Stay safe and healthy. Allah Hafiz.